Уважаемые коллеги, доброе утро.
Dear colleagues, good morning. We continue our dialogue, and today we are discussing a very important topic. Our session is very important. Our yesterday's session, Med Talk, was a step on the way to the discussion which we are going to have today. We have been preparing for these conversations about new medicines, new drugs, new approaches to medical treatment, to cure diseases. Why I'm a moderator of this session, to be honest with you, I have no idea. Actually, probably it happened that AIDS is a very serious reason for all the changes which took part in medical technologies, new medicines, science, approaches. In general, there was a technological explosion due to the huge investment, scientific basis establishment for working with AIDS. Unfortunately, we do not have vaccine. So far, I remember the first patients, we couldn't help them. After that, we obtained some medicines which allowed them to live long. And now we have a set of advanced medicines, advanced drugs, which are not toxic, which allow a person to live good quality life. And of course, it's a great victory, but so far we do not have vaccine. At the same time, we have amazing achievements in study of viruses. I remember 7 years ago or 8 years ago, I was shown a protocol of testing new medicines aimed at hepatitis C cure. And when I saw it, it seemed impossible, because we all know that it's a tender killer. And the measures which we took, all the medical treatment that we provide, is useful for a small number of people, and suddenly it turned out that it's no more a problem. And that would be it, and I would like to give the floor to Andrei Ivashchenko, chairman of the board of directors of Scientific and Practical Center Institute of Physics and Technology. You will probably introduce yourself, and I hope that you will tell us about the programs of your colleagues, which are guests, speakers of our forum. We are looking forward to hear your presentation. Thank you very much, Alexei. Probably before I start my presentation, I would also say some welcoming words, and I would probably Evgeny, as a representative of Moscow, the host city of the forum, could say some words. Thank you very much for Moscow. The 18th Assembly is a very serious step. It's a different level which we achieved, and I really hope that by that we will be able to change our approach to the medicines and to their development. We have more and more medicines, but the problem in the world of medicines and drugs is very simple. The more new medicines we develop, the less accessible they are, because every disease could be divided into 100 small diseases, subdivisions, and each subdivision of the disease requires new medicine, which costs huge amount of money. Another question is how to find the correct position, how to make the medicines more available, accessible for our patients. And we don't have to stop development of new medicines, because everything that's happened in the world and what we have today, for example, in hepatitis, it's a huge achievement which is impossible without new medicines. Thank you very much. I agree. Our session is called Modern Art of New Medicine Development. New medicines, and I would like to show my presentation. I have a number of slides to extend on this subject. Great. Next slide, please. How can I click? Do I have a clicker? Thank you. Our prominent scientist winner of the Nobel Prize, Mr. Ginsburg, in his article, Physical Minimum at the beginning of the 21st century, wrote, where will be a breakthrough from physics, different spheres of physics, theory of strengths. And at the end of this article, he wrote that no matter how we love physics, in the 20th century, it performed its instrumental function, and now it's behind biology, medicine, and the science of about living creatures. And there is an opinion that the 21st century is a century of living systems, which is also confirmed by our IT experts. Bill Gates, answering the question what he would do if he wouldn't be involved in IT, he said that he would be involved in biotechnologies, decoding of genomes, human genomes and microorganisms, provided this wave of technologization, comparison of developed countries for implementation of the results which are provided by science. This is a competition which involves the US, Europe, Japan. Now other countries take part in that, China, South Korea. And in general, it's obvious that the countries which provide technical support of these discoveries will produce the products with the highest added value in the 21st century. If we analyze the segment of the market in general, 70% of the market is medicine and new drugs. 20% is agricultural sphere and 10% for other directions. Development of new medicines, drugs during the last 20, 30 years changed dramatically. Now medicine are made with very high precision, like a key with targeted bio targets. And the cost of development increased significantly. Terms of development also increased. This is a comparison with complexity of molecules of a medicine. Small molecules is like a bicycle. Monoclonal antibodies is like a plane, aircraft. And this movement also includes Russia. Of course, in 2010, a program was started, Pharma 2020, on restoration of pharmaceutical industry. Within this program, there were two stages. The first stage was performed, generical import substitution on the territory of the Russian Federation. More than 2 billion rubles was invested in construction of plants, pharmaceutical plants. And the second step, technological innovation, medicines, is not implemented so far. Nevertheless, probably Russia is one of a few countries which has a capability to produce innovative medicines and drugs. We know that the Soviet Union was good at this manufacturing, and we have modern products developments. We have an accelerator by a bridge which confirms that, and it led, Russian market, it led that to the situation that, according to the program Pharma 2020, the half of the market had to be supplied by our own production, but the actual number is 30%. We cover only 30% of our market. We have generics, which are import substitution, and then we have innovative medicines, and this 70%, each percent is very difficult to substitute, because it's absolutely clear when the core patent is still valid, you have the rights for packaging, distribution, etc. That is why the key important task is innovative import substitution. What happens now? Unfortunately, the developments, the discoveries which we have at universities and institutes, they are published and they go to the West somehow. An innovative cycle is closed there, it's a loop. There are a system of startups, venturing, business, testing of medicines, and after that we receive these innovative medicines back. The key task for us at the present stage, to my point of view, is overcoming of this gap between science and industry which we have at the moment. It's a process which is very profitable, because even 10 or 20% of innovative import substitution is about 300 billion rubles a year in terms of sales. And of course the players who will be involved, who will be responsible for formation of institutes and companies to overcome this gap, they enter a very risky zone. On the other hand, they have a chance in a 5 or 10 year perspective to be based on their own market, to start innovative cycle of pharmaceutical industry. And of course Moscow is a leader of innovative development of Russia, has all the necessary conditions, and Moscow could become a leader in this process. I would like to give the floor to my colleagues, to other speakers. I will show you the last slide, answering the question which was asked by Evgeny. Medicines become very expensive, and there are a lot of them, there are many diseases, and we have to solve that. It's a challenge, but it's a great opportunity. We can overcome this blockbuster model which exists in the world, and in Russia we do not have institutions involved in the process. There should be personalized, customized medicines based on cell technologies, gene technologies, and it seems that here, for Russia, for developing countries such as China, there is a great opportunity. Why? Because it's very similar to the situation with the telephone connection. If we take China, for example, there are no wire telephony in such an extent, because there are smartphones, iPhones, and China is on the next technological stage. We do not need these telephone stanchions to put the cables, wires, and something very similar happens in the sphere of medicine, biosystems. Science develops very fast, and there are new technologies which allow us to cure diseases which were incurable in the past. We can go to the next technological stage. And to conclude my presentation, I would like to emphasize that our own Russian innovative medicines and drugs can be developed, both classical and innovative, and other presentations will prove that. And I would like to give the floor to participants of our accelerator BioBridge, which took place in Vienna last year. You know, more than 300 startups presented their projects, and the best 13 projects went to this accelerator. And some of the startups and developments are presented in our session. I would like to give the floor to Vladimir Kozlov. He has the microphone. It's a graduate of physical faculty and medical faculty, startupper, who will tell us about his projects. Good morning, my name is Vladimir Kozlov. As I was introduced, I will tell you about the project Insight, which was presented in Vienna, which we are actively involved in with our team. And this project is, I would call it, the treatment of diabetes type 1. The next slide, please. Why Insight? Insight. N is the name of my sister, Anna, and this project was called after her because she has diabetes for more than 17 years, diabetes type 1, and she is the person for whom I implement this project. For me, my personal target is to cure my sister, and of course we hope to help any person who needs that, every person who has diabetes type 1. It was not accidental that we started working in the sphere of diabetes. We've been involved in this project for a number of years. We are testing the system, glucose monitoring system, non-invasive, blood testing, which also was tested with my sister Anna, and this year we are planning to start selling this system. You see, it's a clip. It's a device which is placed on your ear, connected with Bluetooth, and it measures your glucose. And about the system Insight, I believe that I should not extend on the problem because everybody knows that diabetes is a very spread disease and many people have this disease. That is why we shall talk about technologies which exist in the current of diabetes, about our competitors, about technologies. First is transplantation, which has a lot of limitations. You need a number of dead donors. The tissue from the dead body is taken, and which makes the process very complicated. Number two is the differential of cells. There are different protocols. Stem cells. It's very complicated. That is why this method is also very difficult to duplicate. What we do is a very reliable technology of curing diabetes type 1. We reprogramming the cells of every person using this system, packets into capsules, permeable for insulin glucose, and implant these capsules. What are the conditions? Technology of programming is more advanced. Now there are more studies, researches on this topic. That is why I believe that it's about to be scaled up. Material for capsules is also developed, manufactured by a number of teams in the world, and methods of implantation are also familiar. There are some teams over the world, and there are some good results. On this slide you can see the market. The market is huge, as far as you understand. Global market is about more than $100 billion. And what are our next steps? We are planning to finish all the testings and to test this method on animals and to have a patent of our technology. that would be it. If you have questions, please, welcome. After the session, probably, it will be more convenient not to take time. Thank you very much. Thank you. We have strict timing. This project will be presented very soon. We have Konstantin Novoselov from Singapore at 9 o'clock. We will ask him about the perspectives and the questions. I would like to give the floor to Konstantin Navakin, who represents the cluster of developers of innovation medicines from Kazan, Moscow.
Very big academic cooperation. Thank you so much. My project is aimed at development of innovative therapy of arthritis and arthrose. This molecule was developed in the institute. And today we have a Skolkovo company within the framework of which we're developing this project. The problem of the skeletal and muscle system and chronic inflammation is really hard. This is associated with pain and suffering. Over 50% of people worldwide have direct preconditions to take non-steroid anti-inflammatory medicine. The market is huge, over $12 billion worldwide. The Russian market is over 50 billion rubles. Unfortunately, all these remedies have limitations in chronic inflammations. Gastro and cardiotoxicity are the main problems. You see the molecule on the slide to the left, and it is protected by a whole number of Russian and foreign patents. When getting into body, it is divided in praxien and pyridoxine. These components have synergetic effects, which I'm about to illustrate. On this slide, you can see that our molecule, even in smaller doses, suppresses chronic inflammation on mice 100% when nanopraxien is successful on 50% even high dosage. Our remedy, our drug, is several times more efficient as a painkiller than naproxen, even in smaller dosage. At the same time that naproxen is one of the strongest painkillers. It's a part of pentalgin, which everybody knows. On this slide, you can see that our drug has a record top safety comparing with others. Being short, it's 100% more safe on key indexes of gastrotoxicity, lethality, and other toxicities comparing to other drugs. To cut the long story short, the results of clinical research say that it overcomes all other competitive painkillers and all other parameters, and international and Russian experts agree on that. We have golden medal, received that last year in Geneva. It overcame many levels Skolkovo expertise. It has become the finalist and awarded on foreign competitions. We are following this project for 10 years already, and we have completed the phase of research, preclinical phase as well, and this year we start the clinical research phase with the support of Skolkovo, and we are looking forward to starting to sell it in 2024. And we're planning to get into clinical research in the foreign countries later on. We invite partners to these processes, and we will be glad to see every cooperation and every interest. Here is my information. You can see it in the bottom. Thank you indeed. Thank you, Konstantin. We know that in Kazan there are many developments. This is only one of them, but there is a whole bouquet of it. Next, I'd like to give the floor to the leader of a project who developed new Comiflu and tested it already in the clinics. Aslan, the first and the second phase of clinical testing, as Alexei was saying, we managed to destroy AIDS, turning it from a death disease to a chronic disease. And the flu virus which is mutating quite fast, say that gives us ground to understand that this is a threat to the whole humanity, and the new molecules which are which are less resistant to the virus are more and more relevant. Good morning, dear colleagues. Thank you for giving me an opportunity to speak. In the framework of this short pitch, I'll tell you about Avivir project. That's a home startup of our group of companies, and within this startup, we are developing original antiviral remedies. And the focus of the main target is flu. Each and every one of us knows about this problem, face this problem, but I would like to underline, to stress that according to the data of the WHO, flu, with all its triviality, is one of the key threats to the humanity and to the health and the lifestyle of a human being. According to WHO, from 10 to 30% of world population are influenced by flu every year, and WHO is forecasting over 1 billion cases, with 5 million aggravations and unfortunately up to 500,000 lethal cases because of the flu. Last year, the former head of WHO announced that there is a new threat of pandemic which can overcome Hispanic flu of the previous century, when the number of deaths of lethal cases amounted to over dozens of millions of people. What do we propose in this situation today? What does humanity have? The class of small chemical inhibitors of neuraminidase. This is the only neuro which people have today for curing the flu. The representatives of this class are running out of patent time. The resistance is growing up. There are side effects in our molecule, which is called 5080. Which is a candidate to this class as next in class, has passed the second class of clinical research. It has competitive efficiency. In the second phase, we observed minimum of side effects, and mainly our molecule is original and has patent protection for the upcoming 15 years. What else would like to state? This is how inhibitor class of neuraminidase looks worldwide. Two first molecules are the Japanese molecules and the well-known molecules of Roche and GlaxoSmithKline, Mifluid and Lenza. This market amounts to $1.5 billion per annum, and it might look strange, but with all the focus on vaccination, this market is growing by 14% every year. We are looking forward to seeing our molecule in its own niche in this class, and will become one of these molecules, and will be good for population. I would like to also focus your attention on the structure of antiviral markets worldwide. Worldwide, the inhibitors of neuraminidase is occupying 40%. In Russia, this share amounts to 6% only, and all the rest difference is covered by so-called immunomodulators, which unfortunately, due to change of names and ideas, are unfortunately very often used to cure flu. Using this platform, I would like to attract your attention to the fact that everyday users, they think that respiratory diseases and flu are the same thing. They are different, but they are cured differently. And I hope this information was useful for you. Thank you so much. Not to take too much time. Thank you. I would like to give the floor to technological entrepreneur, scientist Dmitry Shobolov, who implemented quite successfully, a very important project, a technology of medicine, the production medicine. And now he is into innovation, and now he is involved in innovative molecules development and remedy development, drugs development. Thank you, Andrei Alexandrovich, for presentation. Innovative entrepreneur sounds really good. I would like to present you the project on diabetes. This is creation of a remedy to cure diabetes of second type. Thank you, dear colleagues, for diabetes of the first type. What is the difference between the first and the second one? The second one is elementary and received one. Diabetes are numerous. Over 500 million people have second diabetes. In 2 years, there will be over 600 million people. Quality of our foods is contributing to that, and the lifestyle as well. The total market on the forecast of for 2026 will amount to $64 billion worldwide for diabetes of type 2, which is a good potential for investment in development in this segment. What is the threat of this type 2 diabetes? Well, it's sugar. It went up in the blood, then you reduce it and it looks fine. The problem is that even if you control successfully the level of sugar in your blood, according to the statistics and according to the Diabetologists US Association, in 50 cases there will be neuropathy of a human being. There will be degeneration of neural tissue with all consequences as pain, amputation, and so on and so forth, in 50% of cases, even if you sustainably control the level of sugar in the blood. What does it say? That means that a person will have to cure it somehow, to take extra drugs. Now it is symptomatics, now it is pain killing with various drugs, and then depending on the human being. And this case, a lot of money is invested in creating the special therapy. The problem is that this therapy has to be taken during the whole lifetime, and during the time when the person has diabetes already diagnosed, because these are two sides of the same coin: nerve tissue degradation and sensory loss. This is all the these are all the consequences of the same disease. Our molecules are not unique. This is a peptide. It was created in the Soviet Union in the Institute Ibakharan, when peptide experts were trying to create an analog for bayet, but with prolongation in blood to protect it from the influence of various ferments. And our talented person, Mikhail Ivanovich Titov, added polyoactoarginine tail into it. Now all the molecules are more or less the same molecules on active component, which are added somehow to modify this molecule in order for this molecule to be more prolonged, more sustainable, to penetrate various tissues and so on and so forth. This is basically the same approach, but a bit different extension is added to it with Dr. Orginian. It turned out to be that anti-diabetic effect is the same or even higher than the initial element, and neuropathical conditions were figured out on various cases. These are molecules. The gray is the tail. The gray is the main component, the peptide, and the blue is octoarginic tail. This is schematically demonstrated. This is the earliest phase of preclinical studies. This is a proof of concept which says that the molecule has a potential to be a leader in GLP. All GLP molecules present there probably have the same effect against neuropathy, but it is very weak, very small, and these molecules showing high result. And as GLP-1 is walking across the planet very rapidly, and there is a big competition between these molecule developers. Developing inside this class with some new condition is really serious. Mainly, everybody is aimed at reducing influence on heart, muscle, and so on and so forth. Here you have reduction of diabetical neuropathy. Today we applied to 13 countries. We have patents in 10 countries already. Among them, the US, Canada, European Patent Office, Japan acknowledged our invention. The market is estimated, according to this molecule, up to 2035 in $2.2 billion in condition if it becomes best-in-class molecule. Now we are looking for $160,000 for investment because we have an approved grant. We are considering it not as money we cannot find, but as the announced activity from a side of a partner which will be participating in this project. The project is really hard because clinical research have to be large scale ones. They're really expensive, and unfortunately at the moment none of Russian pharmaceutical companies decided to take part in that, not because they have money, just but because they are not ready to take the burden of these risks. Here you can see the strategy of going out, mainly that's for investors. We are planning in first or second phase of clinical research to get there. Here are the success stories in the recent years in diabetes type 2 and GLP in particular. The Bayet purchases and so on and so forth. Now our team is composed out of 4 people. Mainly we are involved in management of the project. Basically we're not doing anything with our hands. Our main partner is Institute of Biological Chemistry, which is involved in synthesis of biotechnological of the molecule itself, creating the remedy formula, preclinical basis, including the American, Russian ones, and the clinical centers. Our 4 managers have very vast experience in working in this dimension. I was working with Andrei Vashenko in organization of the Company of Technology of Remedies, which were organized in 2007, the framework of development of import substitution of 2020 program, and we developed over 100 generics within this campaign. Last year we sold successfully these generics to one of the key players in Russian markets. I am responsible for over 50 generics and 2 original remedies in my life. Thank you so much for your attention. If you have any questions, do not hesitate to address. Thank you, Dmitry. Indeed, our main partners, our main pharmaceutical players are not really interested in working with Russian developers, but I hope that it is a temporary condition. The situation will change. And in order to conclude the group of startups, I would like to give the floor for like 3 minutes to Andrei Evgenin. He is the head of Student Startup Studio. Many projects, both medical and drug products, appear in Student Startup Studios. It might look strange, but it's a fact. Good afternoon, good morning, dear colleagues. Thank you, Andrei Alexandrovich, for your announcement presentation. I had to have the machine training methods report today for new remedies and drugs. But the topic changed, that's why I will refer to it a little bit and about the artificial intellect, and we'll switch into the new topic. I wanted to say a couple of words about our startup studio. Its main difference from regular chairs that we are working with our students in technological campaigns. They're going to young entrepreneurs and they work in the projects, not as the executors, so to say. They are going to the meetings, they write reports, and they are fully involved in the activity and the life of the company. And now a few words about artificial intellect. As we used to say previously, remedies become more expensive, the number of illnesses and diseases is growing, and new methods appear which help to decrease the cost for fighting these diseases. Recently there was an article of in silico medicine, and they received new inhibitor in 40 days. Usually it takes 2 years, but with new artificial intellect technology, it takes less than 2 months. It is also actively used to analyze text data. One of our projects was done with Himrar. We were analyzing unstructured text publications, and with the help of machine learning, we managed to extract professors from the institutes where they were taught, the money which they received, the molecules which they developed, and to rank it. And we can consider which professors are worth cooperating with and which are not. and the main vectors of development, which are trustworthy and which are not. As for the molecules, this is basically it, and let us switch to the second part. This is a system to optimize the work of AIDS Center. We got involved into it last year. We were working with Birion company. Last year we met the top management of Moscow AIDS Center. We found out that we have mutual interests, and now on the basis of Laboratory of Artificial Intellect of MFT, together with Moscow State Center of AIDS, we are implementing various projects. Now, a few startups are in place, like Dr. Fomin's clinic, which has a very successful system which prevents doctors from performing medical mistakes, so to say, false. We have 5 modules in our system, 2 of them for the doctor and 3 modules for the patient. The modules for the doctors help to route the traffic and helps the doctor to find the proper therapy. And the patient is reminded to take this therapy and monitor his health condition. The first module is intellectual system of routing of patients. When a patient is going to the AIDS center, the patient is going to the doctor is sending him to analysis. He's going to the analysis and do the analysis, and then goes to the doctor. We decided to remove this cycle and to make an application, mobile application, where he will give his claims, and then the system will provide the treatment, and with the results of the system you can go to the doctor directly. After that, with cooperation between various entities and various organizations, you will be able even to make analysis in various places. Then the digital doctor's assistant. When a patient is a first comer, he has to take the therapy, but before entering the doctor's office, he enters his anamnesis in chatbot. He is considered the results of his analysis, of instrumental analysis, laboratory analysis, and initial decision on the therapy is already formed. If the doctor agrees with this decision, then the doctor commission is formed, the protocol is signed, and the commission is appointed really fast. The therapy has to be appointed as soon as possible. That's one of the key issues. This system reduces the paperwork for the doctor because you do not have to write down the protocol manually. And a few words about that, and then I will switch to a different topic. Then the clinical data. Artificial intellect system is good that it can find conditions and consequences when we fail to do so. Moscow Center has a big database of information, and we are planning to process it with this system and have a unique database for Russia and for the world, where we will be capable to find optimal therapy with various initial conditions. Not all the patients are getting everything the same, but a specific patient will have specific therapies, like a personalized therapy for AIDS, for HIV. Then chatbot for the patients, which is answering a typical question, and remedy tracker, which helps not to miss the therapy and evaluates the result and sends this to the doctor for the doctor to understand how the patient is taking this remedy. One more module is integration with other entities when the patient is coming to different hospitals. Our module system will analyze his results and testing, and if there is suspicion for HIV, he sends a request to the AIDS center, and if the result is positive, then not all the employees of the hospital will have this information, only authorized one, and that is a cooperation between various departments, and it can be diagnosed more rapidly and provide for better cure if he is HIV positive. This is basically it. Thank you for your attention.
Thank you very much, Andrei. I would like to give the floor. Now I would like to give the floor to scientists. I would like to give the floor to our guest, Professor Shao, to share his developments in the sphere of AIDS as well.
for your invitation to this very important meeting. I would like to share some of our experience in the novel approach for HIV cure. Thank you. And we all know the HIV field, we have made a lot of progress. Diagnostic and prevention, especially therapy. Antiviral drugs have been started for almost a century after the discovery of penicillin on the 30s of last century. But anti-HIV was hit us. A lot of efforts put on antiviral drugs. So we have already more than 40 anti-HIV drugs. But there are still remaining two scientific challenges has not been met. One is vaccine, another is HIV cure. We have close to 30 million people on treatment, but all of them have to be lifelong treatment. For example, people who got an HIV infection at the age of 20, young people, that's to treat for over a half century. So that's a heavy burden on the person, on his family, or the whole society. So how to get a HIV cure? The HIV cure is coming from this person. They call it a Berlin patient, but actually this Berlin patient is an American patient in San Francisco, but he visited Berlin and got a treatment in Berlin. What he got is he got HIV, he got leukemia. He has to ablate his bone marrow, get a transplant. But the transplanted bone marrow is happened to be a CCR5 delta 32 homozygous bone marrow, which is less than 1% of the pure Caucasian people. The Asian, African people do not have that mutation. So, and then after his treatment, the virus at a very low level, after this ablation of his own bone marrow, got a HIV-resistant bone marrow, he has been cured for HIV for over 10 years. So that's only one patient. But we have another London patient, which is I call half patient, because he has been under control of HIV after the same kind of bone marrow transplantation only for 2 years. So we have to wait and see. And from the Berlin patient, we have developed different strategies to provide a cure. As Alexei mentioned yesterday, that to find the patient, immediately provide treatment, and for the patient reservoir, try to kick the virus out from the reservoir, and then using antivirus, the so-called shock and kill, or kick and kill strategy. And also their immunotherapy, and there also the gene therapy. So that's different approaches. So we have, from this chart, you can see that the typical rebound after you stop using antivirus, what we call antiviral interruption. Usually, within 2 months, the virus got a rebound. And we have like Boston patients kept without rebound for 3 months, 8 months. There is a baby, which is provided treatment a few hours after birth. He lasted for 28 months. But then we have only the Tim Brown, the Berlin patient, kept for over 10 years. And in this field, there's another new drug come out, which is broad neutralizing antibody. It's done by the Vaccine Research Center of NIH, This antibody they call VRCO1. This center was set by the President Clinton to overcome HIV vaccine, but they did not succeed with the HIV vaccine, but they succeeded with this very good antibody who can provide antiviral spectrum for over 90% of the world HIV strains. And they have already using this antibody for HIV cure studies, and also they have done a prevention trial in both the homosexual patients, in 24,000 of them are just phase 3 trial, and also in African women with 1,500. The trial should be concluded by the end of this year. So we can see the results for this prevention trial. In China, from our cohort, Alexei asked how we do with this elite controller and long-term survival. From this cohort, we identified a series of broad neutralizing antibody. This is one of the best. It's a VRCO1 class antibody from the Chinese long-term survival. And we got this antibody. It even has a broader neutralizing spectrum. Like a VRCO1 is 91%. This one is 93%. So we are a little bit broader. They can neutralize the VRCO1 resistant viruses. And also, because we got this antibody in our cohort pretty earlier, so most of the other broad neutralizing antibody, the early event has not been captured. So this one has been, we can use that to study the maturation of the broad neutralizing antibody to see how they evolved and get matured. It can also both for the vaccine work and also for HIV cure work. And then we have a second area is immunotherapy. So we did this on the primate studies in the rhesus macaques. We challenge the virus and get an infection model, and then we start antiviral treatment. During the treatment, we immunize the monkey with our vaccines. And then at the high peak of the vaccination response, we draw blood from the monkey and in vitro stimulate with the HIV antigen and amplified the CD8 to expand them to 10 to the 10th to 10 to the 12th level, and what we call the CTR lines. And then we stop viral therapy, and we get the viral rebound, and we infuse the monkey with the amplified CTL clones, and to see if they can suppress the virus. From this chart, you can see we have a 3 monkey on the upper left corner. The viral level after rebound is very, very high, millions. And at this high viral load, when we give in 10 to the 9th CTL clone, and we cannot control the viral. But on the right upper panel, the 3 monkey with a low level viral rebound, after infuse, we can get a very good control of the virus. They can last 1 monkey. There are some small blips, but the other 2 monkeys are completely in control. So what is the element to cause the rebound virus to be controlled? So we did the study after viral controlled. We bleeded the CD8 cells by using the anti-CD8 antibody, infuse the antibody into the monkey. From the chart, you can see the black curve, which is the CD8 cell goes down. After CD8 cell in the monkey are bleeded, and the virus coming back. So that tells us that the CD8 clones are there to control the virus. If you remove them, the virus get coming back. So this is first time which showed the CD8 infusion, what we call passive immunotherapy, can control the rebound virus in primate model. So that paper was published in Nature Communication. And lastly, the gene therapy. The gene therapy in China was done by the other scientists in my university, and also we have American-trained biophysics, and he's learned to do this sequencing. But in collaboration with American professors, medical professors, they built a company in China, and they did gene editing baby embryos. And so what we call a better practice. So that, I do not see, this is the scientist who did this. His name is Jiankui He, and he conducted on 5 human embryos. He even delivered 2 twin babies. And so after he reported this in Hong Kong in the global international meeting, the government noticed that, and he was caught. And after 1 year investigation, it has been found that he deliberately envisioned the supervision for the HIV infected couples and for their baby, and he forged the ethical review documents. Nobody reviewed, but he made a review. And he breaking our national law of banning gene editing embryos for pregnancy. So it's just 2 weeks ago, he was sentenced to jail for 3 years. He was banned for the lifetime for gene therapy. All the scientific communities are against such a deal, including myself. We have this policy comments in the Lancet against such deal, what we call HIV professionals. We are totally against that. But we also have a very good example of scientists who studied with the patient's stem cells. And the patient, like a Berlin patient, is HIV positive, and he got the leukemia. He has to bone marrow transplantation. But before the bone marrow transplantation, my colleague, Dr. Hong, he edited with the CRISPR technology, edited CCR5. And we can see that after radiation, the patient's blood goes down. After infusing the gene-edited bone marrow stem cells, and then go up, all the blood cells are recovered. And also, this we can see all the blood cells and platelet and all the value goes to normal after they kept that study for over 19 months, close to 2 years. And the most important, they cured the leukemia, but after they stopped using the antiviral interruption, the cells, the virus still got rebound. We can see the A panel, the viral got a rebound. The transplanted stem cells with the CCR5 editing didn't control the virus. The CD4 cells carry the CCR5 ablated during the C panel. They increase their proportion in the blood because there's a virus coming out again. They are killing more the CCR5 positive cells than the negative cell. The negative population get enriched. So this paper was published in New England Journal not long ago, a few months ago, and Dr. Deng, my colleagues in Peking University, he got named as the only Chinese scientist by Nature, the top 10 most influential scientist. Even though the first try is not fully successful, but he made the first try. He showed that you can using this CRISPR technology to edit the bone marrow to provide as a therapy for cure in patients. And there's no side effect about 2 years. So there will be, he's working on to editing more proportion because the total cells is about 5% of the total bone marrow. You need more. He's tried to over 50% of the total bone marrow CD4 cells being without CCR5. But also there's another Chinese scientist published a paper in Nature Medicine, which showed this can be used for typical subclass of patient, but not for general population. The general population, they did a large study of 400,000 Caucasian people in the British cohort. And they showed that by statistics, the homozygous for delta 32 on the two chromosomes without delta 32, with delta 32, there's a 21% increase in the all-cause mortality. So you get a resistance to HIV, you gain something, but you lose something to other diseases. Like CCR5 is an important parameter in our immune response against the pathogen. So without that, your general mortality increased for 21%. It may not be generalized. For HIV eradication or cure or functional cure, we are still at early stage. So to reach the bar, it takes a longer time. So
Excuse me, Professor Shao.
Yeah.
If you don't mind, we maybe make the bring to...
Yes. This is the last slide.
Okay, please.
This is, I'm on
because
the board.
we have the teleconference now with Singapore, so...
Okay, sorry. This is, I'm on the board. We had early last year, we had a meeting. We set the goal for the next 10 years research.
Okay. I think it's really like Alexei says that the HIV is the era where all type of drugs now fighting to come to the cure of this disease, and this really very innovative area and which develop all drug discovery. So thank you very much, and I hope after our teleconference with Singapore with Konstantin Novoselov, we have more time. Professor Volchkov also come and do the CRISPR-Cas9 and gene therapy discovery. So we have more time to discuss because I have also a lot of questions. Thank you.
Thank you very much. Probably our colleagues will provide the connection with the Professor Novoselov, who agreed to join us from Singapore, where he is. I can see Konstantin. Yeah, I can see you. Good morning. Thank you for joining us. As we agreed, I would ask you some questions. First of all, we were talking about science. We are in physics, but our science is on the second stage, as our colleague said, because the biology and medicine is very important science. Do you agree with that? Yes, that's true that biological and medical developments are very fast now, and we have very interesting results. Genome modification, biochemistry is one of the most attractive and fast developing science. At the same time, we should not forget that the main problems in biology and medicine are still there. This science includes very complicated systems, and we do not have the necessary means to describe them as a whole system. That is why I think that the next breakthrough in these spheres will take place on the border of biology, gene engineering, physics, maths, probably chemistry. We need a lot of new methods and means to describe complicated systems, and we absolutely need synergy between different areas of knowledge. Yes, we, being special experts in physics, providing tools so biologists and chemistry experts could apply them. I know that your developments are also related with medical technologies. Could you tell us something about this?
От физиков требуется только разработка.
Methods is about the development of methods and gadgets and devices. It exists as one of the dimensions of my work. I have to say that I have quite a broad spectrum of interests, and we are working a lot on nanoelectronics, on new materials in general. As for biological dimension, I can focus on two dimensions, as you have said. This is nanomedicine and nanomaterials, use of nanomaterials in medical purposes. Rapidly developing the electrodes, biosensors, so on and so forth. The second dimension, is more fundamental. That's what I was speaking about previously. It's an attempt to learn how to describe complicated systems, complex systems. And here we create artificial materials. We call them living materials, life materials, which can think on their own, so to say, or to adjust themselves to the changes of the environment, so somehow simulate the demeanor of the behavior of the living organisms. Thank you. You remember, you probably witnessed Soviet science when you were learning in Fistech and basic chairs, and you perfectly know the international science and the level of development. Can you evaluate somehow the potential of Russia and the science, its perspectives to be built in the international spread of labor and division of labor? Where are our strong parts and where we should enhance significantly the situation, especially in the sphere of technologization, which is happening in these segments? To be frank, what I'm witnessing, I have been witnessing recently, a big number of new technologies appear in biology and in medicine, and still there is no unified technology which definitely wins, like 100% wins. They are numerous and they're all interesting, they're all developing really fast. And from this perspective, we are all in the same conditions. That's why it makes no sense to split it, and we have to develop it in Russia as well. I don't think Russia is falling back or trying to erase it. Within a few years, everybody are all in the same conditions. And I think that people who will learn how to combine the lost results from various segments of knowledge, and in Russia, physics, mathematics developing really fast, I think they will be on top. They will be the leaders in this race, in this chase. And you all know that contemporary ways of looking for new remedies, new drugs, based on big data, on correlations between, looking for correlations in the databases. That's what we can do really good, really well. And I'm convinced that these examples are numerous, and we can find many examples where Russia would be maybe not a leader, but be really close to the leading countries. There were a few startups speaking before you, which you saw in BioBridge 2019, the startups which are in the newest history of Russian science, Russian development of remedies, creating new molecules. I wanted to ask you to share your impressions. You were a part of the BioBridge Scientific Council, which existed in biotechnological sphere. What is your sensation on this event, and how you plan to do so? I have to say that I'm not an expert, obviously, and everything I can share is my impressions. And the impression is that what I was surprised with is a big diversity of what was there. Projects were different both by methodology and by the level of development. Some initial phases, some are ready to produce this, to give the product, to develop the product. It was really impressive for me. And the next is the level. These were really strong scientifically reports and the projects. I remember myself that in Vienna there were really strong professors, and we were talking about, we were discussing on a very good scientific level, and all the projects were, as I saw it, on a very decent level. So it was positively surprised. I think the potential here is really high. Thank you, Konstantin. Thank you indeed. It's really important for us not to feel like outsiders. This look out to you from the top levels of science is really important for us. Thank you so much for joining us, and we hope that many Nobel Prizes will be given to Russia, to Russian scientists as well in top systems. Mano, thank you, thank you. Thank you very much. I'd like to give the floor to Professor Pavel Volodchkov of Russian Moscow Physical Technical Institute, who is involved in editing the genome and gene therapy, to comment on the speeches of Professor Shao, and the discussion which we have had here. Thank you. Thank you. Actually, I did not expect that so many people will be here and so early. Thank you so much for coming. Otherwise, we would have to discuss it only among 5 people. Actually, I liked both the startup reports and the report of our guest from China. And in fact, I want to focus on the last thing, a very complex approach to HIV therapy, a full-scale approach. In fact, Chinese CDC is trying to implement all possible approaches, even the prohibited ones. I'm joking, of course, in order to cure HIV. I have a big number of questions, in fact, both on these antibodies which you use, which were received from, as far as I understand, from people who can get away without highly efficient therapy for quite long. This might be a good hint which will allow to either solo or combining with other treatments to take HIV under control. There are a couple of questions which is really interesting. The article on CTL. I understand that this is a very early work, and at the moment it is tested on monkeys. I also dealt with monkeys, and I know that such works when you deal with CD8 cytotoxic lymphocytes. It's really hard to work with outbred animals. You have to combine them all the time. In fact, like you have CTL, the cell receptors, and in order to have efficient therapy, you actually have to find MHT combinable animal. Maybe due to being limited with the time limits of our reports, maybe I did not understand it quite probably. I have a very big number of questions, but as immunologist, I understand that this is a very efficient way to recruit the immune response with existing antibodies and T-cell receptors in order to fight the immune deficit virus, the human immune deficit virus. As for the editing of various cells, being in Harvard, working as a postdoc in Derrick Rose laboratory, we were one of the first who were using CCR5 editing using CRISPR-9 on stem cells. Obviously, it was edited in the 90s with nuclease on zinc finger nucleators. For example, Sangama company. I have a question to you. Maybe I should better switch into English.
I guess I should speak English because I want to actually ask you about the last part of your talk, when you actually were showing data when you do genome editing of CCR5 at hematopoietic stem cells. And I know it's a really problematic area because like superficially it looks fine, like that should work, but actually the digger we deep, the more problems appears. First of all, it's a problem with virus tropism, so it's not always actually CCR5, and it's a lot of data on this, and data actually trying to repeat this famous experiment with Berlin patient, and it's actually, from what I know, it's more than 10 or maybe 20 already patients was done trying just to repeat the same experiment. It was unsuccessful. And I also know data from Sangama company. It's actually not really that straightforward. They do have very efficient instrument for genome editing of CCR5 locus, but it's actually what they're facing that it's actually really difficult to reach good numbers of cells. Yeah, you do transplantation, bone marrow actually hematopoietic stem cell transplantation, and it actually bring all this gigantic number of problems which is correlate with actual transplantation. So what do you think about perspective of this particular last approach? Should we push it further, or should we try to do something else, like just go from the side? Thank you.
Yes. Yes, thank you for the question. As far as monkey, we all know is a monkey is not a pure animal system. It's like a human. We have diversity. Monkey has diversity. So monkey, we just to do proof of concept. You know, the largest monkey trial is less than 20 per group. And human, you can do several hundred, several thousand. So it's just one step, intermediate step, justified for clinical trials. So we don't totally rely on monkeys. It's like HIV vaccine field. So we have an old saying is that mice liars, monkey do not always tell the truth. So we have to rely on clinical trials, on large number of statistically significance. As for the CCR5 gene therapy, your question is justified. So that is why many groups are doing that. But to be on clinical, to test, Dr. Deng, my colleagues in Peking University, I'm affiliated, I'm a adjunct professor in that university. He is the first to prove. So he has all the problem. You cannot edit at a high proportion. It's only like 8% of the total CD4 cells. But at this level, you cannot control the viral rebound. Too less than 10%. It's not strong enough. But he showed that this kind of another is by safety. You know, if you editing to the target, you can prove. But the shooting gun, you hit some important genes, you don't know. So you have to go on a clinical trial. But his trial said up to 2 years is safe. I think for generally speaking, for HIV, we will not use as a large scale therapy scale. It's only for people who have AIDS, who have leukemia, or have the cancer. Very small subgroup of patients who has that specific needs. So if you don't do, they will die of cancer or leukemia. So that is to provide you an opportunity. But you have to go step by step. It's like antibody. So NIH, VRC01, we know one injection is $10,000. How can you do prevention on tens of millions of people with such expensive drugs? You cannot. But NIH, they did that in 2,500 people, another 1,500 people, 2 trials, already 2 years. But they, in WHO meeting, they showed the one injection, they can reduce the cost from $10,000 to $10. So they have a production line, which is very efficient. They can reduce the price for 3 logs. So everything is we move step by step. But for this gene therapy, it will be only for very few patients, case by case.
Спасибо большое. Я бы хотел бы
Thank you so much. I would like to conclude our session with a question to Alexey Israelevich. When AIDS will be a curable disease? When should we expect it? Bear in mind all this discussion. What should be done? Thank you. Thank you for an opportunity to hold this session within the framework of our Assembly Healthy Moscow. I think that those efforts which we do, which Physical and Technical Institute is doing, the center of Himrad, which is a leader in Russia in development of not only new drugs, but as a leader of has been an intellectual platform for working with that. Basically, this is the precursor to the answer which we are trying to find for 2 years with Professor Shao, with my colleagues, whether we will be able to fight and to win from the AIDS. If we do so, probably the level of breakthrough which will contribute to it will manage to assist to combat certain types of cancer and to win there, and the diseases which today are uncurable. We have to move on, we have to move forward, and it seems to me that Moscow Healthcare here is an ideal platform for such work in this country. You can see this exhibition with your own eyes, and I invite you once more to see the stands, to see the program. Everything is recorded. Our session is recorded as well. Yes, Moscow is a perfect platform which can use in a very up-to-date level to operate and to create the partnership relations to our colleagues worldwide. Thank you so much indeed for Alexandrovich, Professor Vachenko. I'm really grateful. Thank you, dear guests. I'm sorry for having no time for questions, but after this discussion, I think if anybody has any questions, you can come up to the speakers. Have a good day, and thank you so much.